The last talk in this session is by Gabe Nespoli, and like Jyoti, who's just given her Swan Song talk, apparently this is Gabe's Swan Song talk from the Smart Lab, which is Frank Russo's lab in Ryerson, because Gabe's about to finish his PhD, so congratulations for that. And Gabe's going to talk about syncopation and tapping.
Great. Thanks, Matthew. And thanks everyone for coming and watching remotely, whether you're in Graz or maybe at home or on the train. I'm going to talk today about syncopation and tapping and neural entrainment as well. So I guess I'll just jump right in. So what I'm interested in my PhD generally, I guess, is how music makes us move and the neural mechanisms behind how music compels or creates a desire to move. So in the literature, this is called groove. Groove is considered the desire to move. And so one sort of aspect of music that I'm interested to see how it affects movement is syncopation. So this is one of many definitions of syncopation, but it's a rhythm that defies the expectancies created by the meter. So the meter in music is the sort of isochronous temporal structure that underlies a rhythm. And rhythms are often perceived as being laid on top of this metrical structure. And so a syncopation is a rhythm that maybe doesn't fit exactly within that meter, and it kind of defies the expectancies created by it. And that's what a syncopation is. So there are at least two previous studies, one by Maria Witek and one by George Siros, showing that moderate levels of syncopation increase ratings of wanting to move. So if a rhythm is sort of too straight and fits the meter too much, people don't want to move as much as if it's a little bit syncopated. And if the rhythm gets too syncopated and it's sort of too far away from the meter, then people, again, don't want to move as much. So there's this optimal level of syncopation that gives rise to the greatest feelings of wanting to move. And then other studies have found that groove is associated with better sensory motor synchronization or tapping. So Janata et al. In 2012 have found that songs that had higher ratings of groove also had people tap. They had better tapping, tapping that was better synchronized to the meter of the song. So my question generally is, what about neural entrainment? What happens in the brain when neurons are entraining to the rhythm and meter? What happens in high groove conditions? Or in this case, syncopated conditions. I just sort of explained that music provides a great temporal structure for synchronizing movements, and we know that the brain can entrain to the meter of a rhythm. Sylvie Nazardan has a great series of studies on that sort of thing. And then in terms of where in the brain this might be happening, there are again lots of studies showing that music engages the motor system. And I won't get into it too much now, but the temporal structure of music, I feel, suits movement of the body quite well, because motor programs that have developed over the course of evolution are tailored towards the fact that we have two legs and we walk, and other kind of movements like that. And of course, music is often perceived in a duple meter, and so there's groups of twos, and so those things all kind of come together in my mind to create a great pairing between music and movement. And so it's sort of comes as no surprise then that you see activation in motor areas of the brain in response to music. So since groove is this desire to move, then I would hypothesize that if you're listening to music that's high in groove, you would see more activity in the motor system, or more entrainment in the motor system. If you have a greater desire to move in high groove conditions, then you probably have a good idea of where the beat is. You can make good predictions about where the beats are, and so you can move better in concert with the music. So for the current study, I was interested in syncopation specifically, and I hypothesized that moderately syncopated rhythms would give rise to better ratings, tapping, and neural entrainment. So sort of replicating previous work, that I would see higher ratings of wanting to move for optimally syncopated rhythms, also see better tapping performance, and see greater neural entrainment in motor areas of the brain. So the stimuli I used, I obtained from Maria Witek, Thomas Matthews, and Peter Wust. So there were 6 rhythms. I'm showing 3 of them here, 2 each for low, medium, and high syncopation. So the moderately syncopated rhythms were just the sun and rumba clave rhythm. And then for the low syncopation condition, they took those rhythms and sort of pushed them to be more on beat. And for the high syncopation, they moved them to be more off beat. So don't pay attention to the harmony here. Their stimulus set also varied harmony. I just took one of the harmonies, but it's the rhythms that's important. And I'll play through the rhythms here. So this is the low syncopation, I believe. And the moderate syncopation, this is the son clave rhythm. And high syncopation. I also jittered the tempo because this was like more of a control because I didn't want people to just entrain to the same frequency the whole time they were in the booth. And I'll leave it there for now. So the procedure, I had 39 Ryerson undergraduate students participate. They listened to 30, so there were 6 rhythms times 5 tempos is 30 trials. They listened to them 30 seconds each. And they did it in 2 counterbalance blocks. So in one block, it was just passive listening. And the other block, they were tapping along on a MIDI drum pad. I did collect EEG during the tapping block as well. I haven't looked at those data at all yet, but I plan to look at it in the future to see how much motion artifact there is, or maybe there's something else to be gleaned from those data. And then after each trial, I asked them, to what extent does this rhythm make you want to move? So I believe this is similar language that previous studies have used, but essentially getting behavioral ratings. So for the analyses, the rating analyses were pretty simple. I averaged the ratings of wanting to move from both blocks and submitted those to statistical analyses. For tapping, I was looking at tapping variability. So they tapped on the MIDI drum pad. Each tap I aligned to the nearest beat in the rhythm, and the asynchrony was calculated for each of those taps, and then the standard deviation of all those variabilities was my measure of tapping variability for a given trial. The EEG analysis, I'm not going to dig too much into it, but we used a 128 channel biosemi EEG cap. And then I was interested in localized activity because I was looking at activity in motor areas of the brain. I chose to look at auditory and motor to see how they might interact or if there were differences between the two. So to do that, I used an independent components analysis. So basically, an ICA will break down the electrode level data into sort of its component sources, based on those sources' purported contributions to each electrode. And then from those sort of weights of each electrode, you can fit a dipole model to the topography and estimate the center of that dipole in 3D space. And that's what I used to localize each source of activity to see where it was. So for the localization procedure, I used the Talarak daemon from talarak.org. So basically, this is an applet or a command line utility. You can give it a set of Talarak coordinates, and it'll give you back brain regions in Talarak space that it's close to. So in order to select components that were in these different regions, I specified my auditory region as consisting of Brodmann areas 22, 41, and 42, and the premotor region as Brodmann area 6. So for a given participant, any components that had Talarak coordinates close within about 0.8 centimeters to these regions, I assigned that component to that region. If a component was sort of close to 2 regions, I put it to the closest one. What were the other conditions? If there were 2 components that were to the same region, I took the maximum entrainment value, which I'll get into in a second. And if there was no component that was within that distance, then I assigned an entrainment value of 0. Okay. So here's a quick plot of the average dipole locations across all participants. So in blue, we have the premotor region, and in green is the auditory region. And I guess I didn't mention, too, that my measure of I was looking at neural entrainment. And so for the components that were assigned to the regions in order to get the measure of neural entrainment, I just took the time series of the component activation, looked at the FFT, and looked at the beat rate for the given trial. And that was my measure of entrainment. Okay. So for statistics, there were sort of two analyses that I did. One was sort of a standard repeated measures ANOVA with syncopation degree as the within subjects variable. For the EEG data, I additionally had levels of metric level and brain region, so auditory and premotor brain regions. Metric level, I was looking at the beat rate, so the quarter note rate, and then groupings of two, so the half note level, and then subdivisions of the beat, 8th and 16th note levels. And then I also did the inverted U analysis that Witek et al. Did in their 2014 study. And so basically, for each participant's data, I fit both linear and quadratic models to their data and performed an ANOVA on the adjusted R² values with model type as the within subjects variable. So the idea here is that we're looking to see if the data are better fit by a linear or a parabola. And so in her study, she found that a parabola was a better fit to the rating data. So there was basically showing that there was an inverted U relationship between feelings of wanting to move and syncopation degree. And so I would expect to see the same, or I hypothesized that I would see the same thing in my data. So on to the results. So for the ratings of wanting to move, I did replicate what was found in previous studies. So the ANOVA was significant and the inverted U analysis was significant. So a quadratic model explains significantly more variance than a linear model did. So that was all fun and good. For tapping, I did not find that there was this pattern, and instead it was a little bit more linear. So the ANOVA was significant, and the inverted U analysis was not, but we see that as syncopation increases, so does tapping variability. So you get worse at tapping as the syncopation increases. And the neural entrainment data, there were significant effects of syncopation degree and of metric level, and also a significant interaction between the two. So if we just, I'll just zoom in on the beat rate. So this was neural entrainment to the actual beat rate, and post-hoc comparisons found a marginal difference between low and moderate syncopation, where moderate degrees of syncopation actually elicited less neural entrainment. So this was sort of counter to what I had expected, but I have some ideas about that I'll talk about in a second. So a quick summary, ratings of wanting to move, they do have an inverted U relationship with syncopation, so I've replicated previous work. Tapping variability increases with syncopation, so you get worse at tapping as syncopation increases. And neural entrainment to the beat seems to be reduced for optimal levels of syncopation. So why might this be? Why would entrainment decrease when ratings of wanting to move increase? So my interpretation of this, I'm looking at it through the lens of predictive coding that actually Marie Witek and Peter Roos have written about a little bit. So in predictive coding, you sort of think of the brain as a prediction machine. And so it models its sensory input based on previous experience. So the extent to which the input and the model match one another is the extent to which the model fits. So if the model is a perfect fit, then there's no real error between the input and the model. You're doing a good job of modeling the input. If the input gets so complex that there's no sort of existing model in the brain that can model it, then again, there's no prediction error because you're sort of in this state of trying to generate a new model for this input that you can't describe. In between, when the model's sort of a good fit but doesn't fit perfectly, there's a little bit of prediction error there. And the idea is that in predictive coding is that you would then update the model, or Witek et al. Say that there's a string of hierarchical encoding and evaluation that ultimately facilitates feelings of pleasure and a desire to move. So that's in the context of rhythm perception. But the idea is that if the model kind of fits, then you would work to update the model, and that would be an adaptive thing. And so it's been suggested that in the context of rhythm perception, if the model is modeling the rhythm, then there might be this desire to want to move along in an effort to reinforce the model. So if we go back to my data, this is how I'm sort of thinking of it. In the low syncopation condition, you have this scenario where the model and the input match each other really well. And so you get high entrainment because the model is doing really well, and you get relatively lower ratings of wanting to move. And when the model starts to when the input, starts to pull away from the model, there's this drive to reinforce the model, is what I'm suggesting. And so if this model resides in motor cortex, then maybe this emphasis on the model involves moving. So actually carrying out what the model is trying to tell you in an effort to sort of force the rhythm into the structure that you've got. So this is what I'm suggesting is maybe the neural mechanism behind feelings of wanting to move, is that you've got some input that you can kind of describe, and you're trying to make it fit. And so you try to enact those, you try and carry out whatever model you've got going on. And if this model resides in motor areas, then maybe that involves moving. So I guess I've got a little bit of extra time. Okay, so I'll bounce back. So if we go back to the interaction plot, you notice there was also a significant difference on the eighth note level. And so this is a bit more of a simple explanation, but the way I'm thinking of that one is that the pattern of results here kind of mirrors the tapping data. And there are studies finding that there's a subdivision benefit for tapping along. So if you have some rhythm that you're trying to synchronize with, if you subdivide or tap or sort of think about putting a beat in between each beat, then you're better at tapping along to it. And so what seems to be happening in the neural entrainment on the right there is that this is the graph on the right is basically an entrainment to a subdivision of the beat, so entrainment to the eighth note. So for low degrees of syncopation, there's greater entrainment to the subdivision and also better tapping. You have lower variability. And then as the rhythm gets more complex, there's less entrainment to the subdivisions of the beat and your tapping gets worse. And that's it. So thanks to Fran, Joseph, and Emma for helping me collect the data, Thomas, Maria, and Peter for stimuli, and Sean for lots of analysis conversations. Thank you very much.
Thank you very much indeed, Gabe. Questions?
Hi, Gabe. Hey. Obviously, really interested in this work. It's fantastic. A quick question about your metric for neural entrainment to the beat. You said you're putting it into an FFT. Are you just taking the amplitude value at the beat bin? Are you doing any type of normalization across subjects? Can you just sort of walk us through a bit of that?
Yeah, sure. Yeah, I am normalizing within each participant so I can compare the neural entrainment values between each. And I am, for these stimuli, I'm taking the value of the exact bin. In a previous study where I was using natural music, I did take sort of a maximum of a width of bins, but because these were MIDI stimuli, and so I knew where the tempo was, I took the exact value. Yeah.
Thank you very much.
Thanks. Neat stuff. Just a clarification question. When they're tapping, were they tapping the rhythm they were hearing, or were they tapping the underlying beat?
They were tapping to the underlying beat. There were a few participants who, we explained it that way, and there were a few participants who still were sort of tapping around on the rhythm, so we had to go in and say, no, tap like this.
So then maybe I misunderstood, but when you're doing the tapping analysis, I thought you said you were looking at the distance between their tap and the note they were hearing?
Oh, okay. I may have misspoke then. It was to the beat.
To the beat. Okay. The beat, I might have misunderstood.
To the underlying beat, sense. Yeah.
Okay.
All of the tapping was all beats and beats.
Okay.
Tapping to beats and analyzing.
Because for that third one, there were actually hardly any beats that were performed, and it was extremely difficult to even figure out what the metrical structure was there.
Right.
Thanks. It appears that you have the capability potentially to look at entrainment in auditory and motor areas separately. I was wondering if you had done that, if it was a similar pattern you observed.
Right. Okay. So in the ANOVA there was no difference, no effective brain region, so there wasn't any difference between the two, which was a little bit too bad. I don't have the plots here, but there are trends, sort of where you see less entrainment in auditory cortex and more in motor cortex. I don't really know what that means, but yeah, I'll leave it at that. I mean, this is a relatively new analysis, and I think it's pretty fuzzy. I mean, the resolution is obviously very coarse, but I'm hoping that if you're just doing a few brain regions and they're sort of sufficiently far away from each other, and you try and keep things going, then you can kind of get an idea. But I mean, I was talking to Charlie the other day about using higher density EEG, and you can use MRI to help you guide the localization of those things. So I think this field is probably going to get really exciting in the next little while.
Do we have a question from Graz?
Is that what he's telling us?
I think there seems some sort of lag going on at the moment. Okay, that's fine. Yeah. We hear you. Well, we see you. We don't hear you.
Thanks a lot. Just a bit of a technical question. I'm trying to understand how you did the dipole fitting with independent components analysis. Okay. Did you do any kind of dimensionality reduction before you performed the ICA, or did you get 128 different sources, and how did you choose them? I've tried to do this before and struggled with it, so I'm interested in how you did this.
Yeah. Okay. I mean, it's not. I was going to say it's not an exact science, but I was using EEG Lab to do a lot of it, and so I did what's done in there. I believe there's some PCA reduction that happens before you run the ISPA. I did get out from the 128 channels minus dirty channels, over 100 components per participant. I was also using a residual variance threshold on the dipole model. So if the dipole model, if the sort of the explained variance of the dipole model was 85% or greater, I only considered those components when I was selecting them for localization. So I sort of required that they were dipolar, basically, and they weren't sort of a messy topographical map. There's previous work showing that the way a single source in the brain would project onto the scalp is dipolar. So you're not going to get sort of colors everywhere. It's going to be a nice blue end and a nice red end.
So within participant, did you choose one dipole for the region that you thought it was, or did you choose several and average them? Or
no. So I was trying to throw a heavy brick at the problem and get the most out of it as I could. Okay. So it was basically only those two criteria. So the residual variance threshold, and then being within 0.8 centimeters of the region. That value is sort of a function of the tool I was using, and I can get into that if you want. But so I basically just threw the components at it and said, these are the two criteria, what goes where. And if there were multiple components to the same region, then I took the highest entrainment value, heavy brick. If there was a component that was assigned to both regions, then I took which one it was closest to, because there's a little bit of a distance measure you can get from this tool. And there were a few cases where it was the same distance from both, at least what I got, and so I just let it be assigned to both. There weren't that many cases of that, though.
That's great.
And then the last case, if there wasn't a component that was found within that distance to a region, then I assigned a value of 0. I assumed that there was no entrainment happening in that region.
Thanks a lot, Dan. That's helpful to know. Thank you.
I was just wondering if your intuition is that, given that it looks like there's less neural entrainment in this condition where we feel like we want to move, if people are allowed the opportunity to move, do you think the neural entrainment is going to be different later, or that's kind of a constant relationship?
Yeah, that's a good question. I think that there's this idea that syncopation makes you want to move because it makes you want to fill in the gaps that are missing, and maybe that would be true. I mean, if you start moving along, then I think I would expect that you'd see more entrainment at the beat frequency. Because if only because you're sort of emphasizing the model, and now you've got sort of more proprioceptive feedback, and you're sort of sort of emphasizing entrainment at that frequency. Whether you would get greater entrainment because for other reasons, I don't know, but I think it would be higher, and I think that would probably be the reason.
Yep. Thanks.
you. The complete opposite end of the design from what Andrew was saying, going to the stimulus side. I don't know about my fellow listeners, but I felt that the easy model was really easy. The syncopated model was really pretty easy, and then I completely lost it, at least in my short exposure to the hard model. That is, I felt subjectively I didn't even have a model to have a prediction error. And I'm wondering if it might be perhaps of interest to have a level between that is challenging, but still allowed the non-professional percussionist to at least have a model to and therefore the prediction. Yeah, I'm sure you did this. Oh, thank you, Michael. Makes me feel better. But I think it might actually be interesting because some of it's not clear to me that was a continuous range of stimuli. It seemed like 2 and 1, and were it possible to generate a level in between? I think there might be some interesting possibilities to look at a prediction model that's challenging but doable. And I suspect you had to throw out a number of trials where people just stopped tapping, or the asymmetry was so big that you lost some data on that hardest one, I'm guessing.
That's a really good comment. And I didn't mention this, but there are ways of quantifying syncopation, and that's actually how I was able to do the inverted U analysis. And so I used the Longut, Higgins, and Lee method of quantifying it. And so the values for the low syncopation, they were both 0. For the medium syncopation, I think it was 4 and 5, and for the high syncopation, it was like 12 and 15 or something. So there is this discontinuity there. I think that's a great suggestion, because I mean those clave rhythms are so ubiquitous as well. I feel like everyone, even if it's syncopated, like you just know it, and so you're able to find the beat rather quickly.
Actually, Gabe, following on from Andrew's question, did you give a metrical framework before the stimuli started?
How do you mean? Like
did you say like 1, 2, 3, 4, and then stimulus starts?
No, I didn't. I just played it and told them to tap.
Okay, if you didn't, just one thing about the first stimuli, which is supposed to have that syncopation value of 0, you could actually feel it in 3, and if you do, it becomes syncopated. Now, most people probably default to some sort of duple interpretation, but you may have some oddballs in there who like to feel waltz with everything. And so
I'm not one of them, so I wouldn't pick that up.
It's just one of those things that maybe that just to provide the model, just the metrical structure to start with. I was going to ask a question about your tapping and entrainment slide, because there wasn't a significant difference in tapping entrainment between the low syncopation and moderate syncopation, but yet you said you inferred an inverted U from the data, and I didn't quite see that. Here? We can't see the slides.
Oh, so
you did revert.
The neural entrainment, you mean? The neural entrainment to the beat?
You did revert to it. Is it possible to go back to the slides or not?
This one here?
No, not that one. Earlier one. That one there. These. Is it the what's on the Y? It's the
tapping variability.
Tapping variability. There's no tapping variability between the low and moderate syncopated conditions.
Right.
So that doesn't seem to suggest an inverted U, does it?
I wasn't suggesting that.
Right.
That was the ratings. I got the inverted U for the tapping. I'm seeing it more as linear. As syncopation increases, tapping variability increases.
Cool. Excellent.
And I mean, this sort of, to Andrea's point, this sort of mirrors the syncopation index of them, where these two are, 0, 4, and 15.
Okay, I think we have time for one, maybe 2 questions.
Thank you very much for your talk. I was just thinking, you mentioned that you used more naturalistic stimuli and had to use a wider bin for the FFT. I was wondering if you also used a more naturalistic movement condition, and whether if you just allowed participants to move however they wanted freely to real music, you might actually find this inverted U, where they move more to music that makes them want to move more, but not necessarily by tapping.
Right, as opposed to this.
Yeah,
exactly.
exactly.
That's a good suggestion, actually.
Although,
I don't know.
of course, you can do EEG if you just ask them to like move around to music.
Right.
But I was just wondering, did you use any more naturalistic tapping conditions as well?
No, I haven't. In that previous
conditions.
study I was talking about, I didn't have a tapping condition, unfortunately,
okay.
so I wasn't looking at it. But
Okay.
yeah, no, that's a good suggestion.
Okay, thanks.
Any more questions? I think that's one more. Coffee will have to wait.
Sorry about that. So thank you for the great talk. So I have a question about the calculation of tapping variability. So if I understood correctly, so you calculated the variability from the isochronous times, right?
Yeah.
So I was just wondering if there were any systematic for high syncopated condition, maybe there were any systematic deviation from the notated isochronous beat times. So if you calculate the tapping times at different beat positions within the one repetition, so if there were people were consistently behind the beat position 2 or only in beat position 3, then maybe if you calculated the variability relative to that mean of tapping, not the metrical grid. So then
I didn't look at any of that. That's a great suggestion.
Yeah. So for syncopated rhythms, maybe we have some kind of trajectory of at some beat position we are ahead, and then in other positions we are behind. That kind of orbit work.
But it's still consistent across.
Yeah.
The
So
trial.
yeah.
Okay. Yeah. No, that's great. Yeah, the whole idea of syncopation, sort of pulling away from the meter a little bit. I feel like it's sort of a continuum in time. As the rhythm unfolds, it might pull away more at some points and push back at other points. So yeah, thank you.
Okay, thank you very much indeed, and particularly thank you to the audience for asking great questions and stimulating a good discussion. Thanks very much.